So what I'd like to do this evening is really to tell you a story, and it's a story about my process of becoming a scientist, a researcher, a mentor, a teacher at Fresno State. And so I've titled it to talk about serendipity, discovery, and joy in chemistry. And the joy in chemistry is really not, meant to distinguish what has happened throughout my life and how I've become a chemist along that pathway. So as Puneet mentioned in my biography, I'm born and raised in Fresno, California. And so what I'm going to tell you a lot about is what influenced me growing up and how I became a chemist and why I became a chemist. And I'll intersplice this story with talking about my mentors and teachers and family and things that have really influenced me over the last couple of decades or so. Okay. So chemistry, people, when you say the word chemistry and that you're a chemist and you study chemistry, you're teaching courses, they often think of it as very challenging and not necessarily with the best adjectives to describe it, but I'm here to define a little bit more about what chemistry is and how it has a benefit towards society and how I feel like when I'm teaching and mentoring students, I want to show them the benefits of communicating science correctly and making sure we explain why research is so important in this world today. So it's really the study of how the state of matter and how it combines, reacts, interacts, and changes. And those are just a few descriptors. But really, when you define it, when you're taking Gen Chem, it's something about looking at what happens between reactants and how they become products. And so in that same parallel way, I want to tell you a little bit about me. So the little joy becoming the big joy. And it's really this arrow right here that makes a huge difference, right? So if you're a chemist, you know that there are atoms, molecules, elements, things that change and go from one thing to another. So in that exact same parallel way, right, there are things that have been catalysts, people that have changed me, things that have influenced me. And yet, because we know with this arrow that you form these products, in real life, right, we know that those things don't always go to the final product. There might be variations of that. And so I'm here to talk to you about those variations and what influenced me. So I grew up in a household, youngest of 3 kids, 2 older brothers in the 70s. And this is sort of my first introduction to chemistry. And you'll see, some of you will recognize, this is the Gilbert Fun with Chemistry kit. And so I was early influenced by chemistry by looking at these chemistry kits that had all these chemicals in there. And you'll see that this is the image that I first saw of what a chemist should do, right? That there's this spark of electricity here, that you observe things in a test tube, that there's this wonderful, colorful reaction that takes place. There's submarines. And it's just this wonderful picture from the 70s of what this Gilbert chemistry kit embodied. This is also my first view of what a chemistry lab looked like and who was in a chemistry lab. And it's just kind of fun to see that, despite the fact that when I looked at the picture of what chemists look like and chemistry labs look like now, it's quite different. Lab coats are still around, lab benches are still around, but the group of diverse people in that laboratory has now changed quite a bit. It's very international. It's very stratified by various female and male scientists. So it's nice to see that there has been some change in this picture of chemistry. But what really influenced me? What got me interested in chemistry the most? It was really this idea of looking at what happens when you think about color and chemistry. And so this is a picture here, same thing in that Gilbert handbook manual, where you have these 2 kids that are looking at this rainbow, and then there's this chemical world that's out there. And so the thing that really influenced me when I was a child was the idea of looking at fireworks and these beautiful reactions that happen when you saw the 4th of July fireworks or any celebratory event where you had fireworks. And that really just enthralled me, enthralled my imagination, and made me very curious in discovering how these reactions occurred, why they occurred, and how they could be influenced. So that was my early introduction to chemistry. 4th grade comes along, and I'm in my public school, Daily Elementary, and our first project was to bring in a piece of food and to put it into a baby food jar and observe the changes. So I, knowing that I had ham and cheese sandwiches every day, brought in a piece of cheese. And I brought in this Kraft piece of cheese, put it into my baby food jar, and just like all my other classmates, every day observed it for 3 weeks, writing down observations of what happened. So I was just ready and prepared to write these copious notes of what I was seeing. To my surprise, but maybe not to others, this piece of cheese sat in that baby food jar for weeks, and nothing changed. And I got very creative in my description of how that cheese melted, oozed, changed form, but really never saw any biological change as far as mold growth, which is what the whole entire experiment was about. But it really perked my interest, right? Because I was being a scientist at that point, observing changes and trying to figure out what was going on. Why did this piece of cheese not change? So, go a few more years ahead, go to junior high, and I started learning from a wonderful group of scientists and teachers at Edison Computech, and I learned all about the periodic table of elements. So anybody who's a chemist knows this periodic table of elements very well. It's sort of our handbook of all the various different chemicals that are out there, and I'll note to the chemists that are here, I've included the newest elements that have been found in the last year, and their names and their representations. But it's really this as sort of the roadmap that helped me discover what chemistry was all about. And it was very serendipitous in trying to figure out, okay, so now that I know all these elements and I can look at these symbols and understand what they mean, what does this truly mean in terms of what I can do to expand my knowledge? Okay? So this is where, when I think about when I'm teaching biochemistry to students in my course, I think about how can I relate this to everyday life? How can I relate it to things that they'd be able to enjoy, that they may see later on in life, where they may find some discoveries that would help them? So this is where I begin my story about biochem. Right? So we've gone from looking at elements, atoms, and now we're going to start expanding that view by thinking of things in a macromolecular structural view. So proteins, DNA, all the things that I've taught Puneet and the other classmates she has in Chem 155A and beyond. So here's our translation of what goes on in biochem. And the metaphor that I draw is that we think about DNA, RNA, and protein. So the central dogma in biochem is going DNA, transcription, translation, RNA to protein or enzyme. And so when we think about the different words and things that make up the bits and parts, I think about them as nucleic acids or the DNA, RNA, representing the alphabet, okay? And then when you look at proteins, you also have things that represent the alphabet. So these are looking at the fundamental bits and pieces, so the nucleotides and the amino acids that make up the alphabet for the nucleic acids of DNA, RNA, and proteins and enzymes. Okay? So this is the fundamental basis by which we explain things related to biochemistry and macromolecules. The words that you form are these codons, so these bits of nucleotides or these amino acids form these words when they come together. And so you can see this codon here for nucleic acids. Likewise, for amino acids, when they come together to form these words, they form these different words that have different structures. And then finally, sentences. So when you take the alphabet, the words, and the sentences, it translates to the nucleotides, the codons, and the genes. Likewise, for proteins, you get your amino acids, your alpha helices, your beta strands, and your proteins. Okay? So this is the fundamental part of what I teach in biochem structure. And one of the things I often do with my students is I like to bring in demos and models. Puneet mentioned this. So these should look familiar to anybody who's had my biochem class. So this is what we call a tuber. This represents the string of amino acids and the polypeptide structure of what you can form from a protein or an enzyme. And so we play around with looking at these structures of how you get all these twists and turns that form a macromolecule, such as a protein or an enzyme. Likewise, I like to bring in structures. Students often will build a paper molecule of this, but this is green fluorescent protein, which amazingly has won the Nobel Prize because it's a wonderful tool for visualizing things that you normally cannot see. So this comes and was isolated originally from jellyfish, but it's a protein that fluoresces green that you can put into other things. And I'll talk about those other things near the end as we go along. So this story leads us, after this description of the alphabet, the words and sentences, to talking about macromolecular structures. So my scientific life, so to speak, has been really dictated by three of these major proteins or enzymes. So I'm going to talk a little bit about each one of these. So the first is copper zinc superoxide dismutase. It is an enzyme that's related to Lou Gehrig's disease, and I'm going to talk about how that was my first protein love when I started graduate school, and I characterized it to find out how it bound up to metal ions and how it reacted in vivo in a biological system. Second protein is called amyloid precursor protein, and it's related to Alzheimer's disease. I really studied this protein during my postdoctoral work and research. And then finally, what I've brought to Fresno State most recently is this discussion of an environmental putative toxin called BMAA that's related to a disease called ALS-PDC. So I'm going to walk you through these three different enzymes, tell you a little bit more about them and why I got so interested in studying them and the discovery process that occurred. So much of what got me into science was thinking about the unknown, discovery things, and really being focused on what is research. This is the experience that I like to tell students about the most because this is something that you don't necessarily see in textbooks or read about per se, but it's an experience that I think every undergraduate and graduate student should experience here at Fresno State. As an undergraduate STEM major, it's really important to be able to go beyond what you're learning in the lecture and expand that into something where you really, truly are discovering something novel that has an impact. And so research to me has been a way in which I can work with students, mentor students, but also translate what they see in the textbook into something that's more viable and more relatable. So what is research? You can think of it as a careful or diligent search, something that's a studious inquiry or examination. But it's really collecting information about a particular subject. And I know that sounds very generic, but once you decide and you figure out that there's a particular area that you're interested in, delving into that subject and really gathering all that information is really what drove me to want to pursue research and pursue an advanced degree in chemistry. So I'm going to tell you a little bit about what we've been doing in our research lab, which is the problem, why we're trying to solve it, and the what, why, and how of it. So I often frame this same type of question to my students when I'm talking about things in the classroom, so that they start to think about it in terms of the way in which a scientist thinks about things. So the what is neurodegenerative diseases. So we know that there's Alzheimer's, Parkinson's, and ALS. Those are all three different types of neurodegenerative diseases. The why is it so prevalent in our society and world? Okay. And the how is, how can we study that? Are there biochemical models at the atomic, molecular, and organismic level? And so these, again, are the way in which I translate the more complicated concepts into something that is able to be broken down into steps and pieces that students can help to contribute to. And I have many students in my research lab right now, and I'll tell you a little bit about their research as we go along. And the who, right? So my research group. So the key question has always been, how can science help a person? And I guess that's part of my motivation for going into research. What is it that I discover that I can contribute to society? What are the things that are most important to me? And part of the reason why I'm here at Fresno State is not only to be a good teacher and a good scientist, but to share that knowledge with others. So this is your Chemistry 101. We're all aerobic organisms. So in other words, we all breathe in oxygen. One of the key reactions in chemistry is a redox reaction, where there's a transfer of electrons and there's a transfer of matter, and you get a reformation of things. So this is an example of what we call 4-electron reduction. In other words, you start with oxygen and you end up with water, and there are 4 electrons here, 1, 2, 3, and 4. That causes the conversion of oxygen to superoxide anion, which is considered a radical, hydrogen peroxide, which is a strong oxidizer, water, another radical called hydroxyl radical, and then finally water down here. So you would think it's pretty innocuous that you're starting out with oxygen, which we all need, and you end up with water. But, as you all know, a lot of times in chemistry, it's the things that happen between the reactant and the product that make a big difference. So this area right in here is very important to study, because oftentimes we note that things that are radicals that react very quickly or rapidly are things that can cause damage to a biological system. So the very first protein I started studying when I was a graduate student was this one here. It's called copper zinc superoxide dismutase. And the main part to note here is that it contains these metal ions, copper and zinc. So this is showing a blown up view of this area right here. It's considered a dimeric protein because you have two parts to it, and you can put a mirror right in here and make a fold, and so it'll fold onto itself. So that's why you call it a dimeric protein. It's a very special enzyme. You can find it in many different organisms, all the way from prokaryotes, eukaryotes. Just about any living organism contains some form of this copper zinc SOD. So its main function is to act as an antioxidant. So where have we heard of antioxidants? Well, we know that a lot of these antioxidants have been proponents and found in various different foods. You have your dark chocolates, you have your blueberries with natural sources of antioxidants. They don't all contain SOD per se, but they contain things that help to reduce and get rid of those noxious species that I mentioned, the radicals. Okay? So SOD is one of those species that gets rid of these radicals. So how does this relate to neurodegeneration? In the early 1990s, there was a connection made between Lou Gehrig's disease, which I'll talk about right now, stands for amyotrophic lateral sclerosis. It's a type of neurodegenerative disease or motor neuron disease. It affects people anywhere from 5% to 95%. There are two different types. And a lot of times when we're studying diseases, there's these two forms, the genetic form and what we call the sporadic form. The idea as a scientist is to study the form that can make the most contribution. So oftentimes we'll study the genetically associated form. But as we'll note, with many of the diseases, they're mostly sporadic. So there's no known direct cause related to the disease. So we have to look at various different aspects in order to figure out what's going on with the disease. There are only about 5,000 people diagnosed per year with ALS, but it commonly strikes people between 40 to 60 years of age. And it's so named because of the famous New York Yankee ball player, Lou Gehrig, that suffered from this disease. On average, this is an especially devastating disease because once one person shows a symptom, they only survive 3 to 5 years after the onset of the disease. And many of you may have heard about this disease. It's often called an orphan disease, but now we hear so much about it because of the Ice Bucket Challenge that was raised a couple years ago. So my introduction to studying biomedical problems related to neurodegeneration started with looking at copper zinc SOD and ALS. So that was during my graduate school time. So I was really focused on studying proteins and enzymes, things that you could not necessarily see in a test tube, but were things that you can characterize chemically. Okay? At that time when I was a graduate student, we had some really interesting research findings. But I found myself in a quandary because I often received letters from patients suffering from ALS and or their loved ones suffering from ALS. And their story was really to write me a letter indicating, the person's life and perhaps some way in which my discoveries and research could help that person. I knew at that point as a graduate student my findings were limited because as a scientist we can only say so much about our research findings. So I expanded my view of what I could study as far as looking at proteins and enzymes to looking at things in a different system, a different model system. So I began my focus on Alzheimer's disease when I started my postdoctoral research in Boston. So I'm going to tell you a little bit about Alzheimer's disease. And many of you either know somebody who suffers from the disease or passed away from the disease, but it's a very prevalent neurodegenerative disease in the United States and in the world. So on average, there are 5.2 million Americans of all ages that have Alzheimer's disease, and that was in the year 2013. 1 in 9 people age 65 plus has Alzheimer's disease. So we know it's a disease that affects the aging population. If we look at it worldwide, as of 2010, there are 36 million people worldwide that suffer from the disease. 66 million is expected by 2030, 115 million by the year 2050. So knowing that this is such a devastating disease that will affect many, many people worldwide, I wanted to study a little bit more about what was going on with this disease. We know that anything that we study in the sciences also has an economic impact as well. So this is looking at the change in the number of deaths in the last decade versus the estimated cost of Alzheimer's disease. And we know that it's going to increase significantly. While a lot of research dollars and funding has gone towards supporting many of these diseases, which is wonderful because we've seen a drastic decrease in these types of diseases, the research dollars funding Alzheimer's disease has not been as high. And so you can see that the number of deaths related to Alzheimer's disease has grown significantly based on the aging population and the fact that we still haven't found any cures or anything to help slow down or delay the disease. So when we look at the cost of Alzheimer's, we know that it's going to cost quite a bit. And it's not only the economic cost, but we know that there's a great emotional cost for the family members and the people who have to help those family members that suffer from the disease. So what's going on with Alzheimer's? This is a little description of the neurology that goes on with Alzheimer's. So this is a normal brain here, showing the sagittal sectioning of what a normal healthy brain looks like. And you can see that there's density of material in this brain, whereas when you look at an Alzheimer's brain, this is a depiction of what it looks like. So there's a lot of shrinkage in the brain here. Okay. So why does that happen? Part of that is related to looking at proteins and enzymes that may affect this shrinkage in the brain. Okay. We know that the pathology of neurodegenerative disease is something that's been well characterized. And so we can think of it again in the protein or enzyme function. And so there are things called plaques that we know affect Alzheimer's, as well as tangles that affect Alzheimer's. So once I had finished studying copper, zinc, SOD, and ALS, I transitioned into looking at the protein related to Alzheimer's disease because I knew that had a very devastating effect on the world population. There's a great group of scientists, not only in Boston, but all over the world studying this particular protein. So these are an example of what the plaques look like and a visualization of what they look like. We know that they build up between nerve cells, and they're made up of these small little bits of protein. So remember when I was talking about the translation of looking at the letters in the alphabet, words, and sentences? These small bits of proteins are like little words that are accumulating together. Okay? So they're forming these clusters of words that shouldn't be where they are in the brain. Tangles are also a proteinaceous material, so they're made up of proteins as well. And so you have these two different types of proteins that form, both what we call fibrils and what we call plaques. And so my goal in studying this, as well as others, was to look at how did these plaques form. So this is for the formation of these ill-formed proteins or plaques, these words that are coming together that shouldn't be coming together at the wrong time and place. And they're formed from a longer molecule. So this is where I bring back my tuber. Okay. So this, again, think about this as the long structure of this macromolecular protein molecule called amyloid precursor protein. It is normally cut. Okay, you find it in the cell interior partially, as well as in the cytoplasm, as the extracellular space as well. We have enzymes that don't look like scissors, but cut like scissors, and so they'll cut that longer piece of APP into bits. If they're not cleared away right away, they do form these plaques. So my explanation of SOD and ALS, Alzheimer's disease, and amyloid beta peptide are two examples in which proteins and how they're processed can make a big difference in neurodegeneration. And so I can bring together things related to looking at not only Alzheimer's, Parkinson's, ALS, and ALS-PDC. So much of the discovery of what we've done here on campus is related to this disease called ALS-PDC. So what are the causes of all these various diseases? So despite the fact that in the trajectory of my career pathway I've studied these different diseases, they're all somewhat interrelated, okay? And I want to explain a little bit about that. So what are these factors? We know that age is a factor that affects neurodegenerative disease. We know that gender can often be influenced. There's a propensity for differences in hormonal levels to affect these diseases. We know that in the central dogma of DNA, RNA to protein, that proteins or things related to each of these different diseases has a protein related to them. And then lastly, lifestyle. So we're not quite sure, but we know that lifestyle can have an effect on the development of these neurodegenerative diseases. So what is the exact story of what's going on? So I mentioned to you, proteins must fold in a certain way to be normally functional, okay? And so this is an example of what a normal protein or oligomer might look like in its shape. So this is the normal form. If they're forming into things that might have a different structure, they become a little bit more complicated. In this case, there are 5 parts to it. It forms what we call a pentamer, has a central area here that doesn't like water. And then we have another form here that's called a fibril that forms sort of this hairpin loop. And each one of these bits and parts, again, are like the alphabet, forming the words, forming these longer sentences. But they can take on different structures. They can even form these things that are a little bit more what we call mature fibrils that look a little bit more complicated. Okay? So this whole idea of protein folding and the way in which things normally fold is very important. So this brings me to the last part of what I wanted to talk about, which is related to looking at the story of ALS-PDC, which is what we've been discovering and working on here at Fresno State. So the story is really one that began on the island of Guam. Historically, in the 1950s, there's a really high preponderance of this disease called ALS-PDC. And it's called ALS-PDC because it shows forms that look like both Lou Gehrig's in ALS, parts that look like Parkinsonism, as well as Alzheimer's disease. So there's a little bit of all 3 of these diseases in this one disease. And so the preponderance of this disease is that it's 50 to 100 times higher on this particular island initially in the 50s than any other parts of the world. So that made us wonder as scientists what was going on here. We also noticed that the age of the onset of the disease was much higher compared to these other neurodegenerative diseases. So that was a really interesting question for us to look at as scientists. So as I sort of jump forward to what other scientists have done, the idea was that we had influences of the lifestyle. So on this particular island, we knew that there are cyanobacteria, as there are cyanobacteria all over the world in brackish waters, in the oceans, in the soil, etc. We know that these cyanobacteria produce a molecule, and I'll talk about that molecule, but it's a small molecule called BMAA. It is that small bit, just like an alphabet letter, but it's an unusual alphabet letter that we don't normally use in humans. But it's produced by bacteria, can be found in this seed of a cycad plant. It's been found in flying foxes, it's been found in benthic fish, blue crabs, mussels, other various products throughout the world. And the idea is that ALS-PDC is related to biomagnification. And this is something that I often do, as Puneet mentioned, that I like to bring in real-world research articles for students to read. So this is one of the articles that we've often read in my biochem classes. But it's one that talks about what happens when you can measure things chemically and then study them for their biochemical and biological effect. So this is looking at 2 forms of this small little alphabet that's not normal, what we call the free form of the BMAA, and one that might be part of a larger word, protein-associated BMAA. So as you go up the food chain to the people, you're seeing that there's more of the small, unusual alphabet letter than you would find in a normal situation. So this is the chemistry showing what happens to this reaction, and I did want to show a little bit about the reaction that can occur. You can start off with this molecule called BMAA that's produced by cyanobacteria, and it can form a variety of different species. Okay, so this double arrow means that it can go back and forth in this reaction. So a lot of times in chemistry we have reversible reactions that can form one thing and then go back and forth, back and forth. So this particular BMAA can form what's called the free BMAA as well as what we call carbamylated BMAA. This particular species can look like another bit of a word that we commonly use in humans. This word is an amino acid that's called glutamate. And so we see some similarities in the structural makeup of what we call carbamylated BMAA with this glutamate molecule. So how do we study this? This has led to what was the final phase of what I've studied in my research career, which is studying proteins and enzymes in a test tube, studying them in neuronal cells, and then finally looking at things that are actual animal models or organisms. And so as a chemist coming from that background, I didn't necessarily want to work with vertebrates, but there are various different animal models out there. I wanted to introduce the audience to this. You can have something as simple as a yeast. So Saccharomyces cerevisiae, which is the same thing that produces the yeast that we use in beer and breads. That can be used as a model system for eukaryotic, human-like system, believe it or not. You can have these C. elegans worms that I know that some of the biologists in our college are studying. You have your mice, your mouse, rats. You can have rabbits. You can have primates. But at the center of my research is this small insect. It's only 2 to 3 millimeters long. It's called Drosophila melanogaster, also known as the common fruit fly. So I know you've all seen those probably at home at some point if you had any fruit that had been out for a little bit too long. So this is the model system that we use in our laboratory. The reason why we love studying it so much is because it's been studied and characterized for so many years. We now know that you can look at the network of all the neurons in a fruit fly and associate that with particular behaviors and things that go wrong in a fruit fly and translate that to what's going on in a human. Because the different types of neurons in a fruit fly are the same types of neurons in a human being. So this is the point at which oftentimes in my classroom I'll stop and I'll get students to talk to each other. And I can't see all of you out there, but I know that in this case you are my students. So I'm going to give you 30 seconds to read this and compare your answer with your neighbor. All right, so normally I have students vote with these cards that say A, B, C, and D, but because I can't see you, I'm going to go straight to the answer. But I can see some of you, so we'll have you raise your hands just to get some audience participation here. So how many of you think that it takes 1 day for a fruit fly to develop from egg to full adult? Okay, I see a few hands. 5 days. Another few hands. 10 days? Okay, 1 month? Okay, depends on the environment. Okay, so there's good variation of answers here. And as with most of my questions, I often have multiple answers. So in this case, C is the answer if you're looking at things in a typical laboratory environment where it's kept with light-dark cycle, kept at constant temperature. And E depends on the environment is also a good answer too, because you can speed up or slow down the development of a fruit fly depending on the temperature at which you raise these fruit flies. So the best part of this model system is that they're very easy to culture. So 10 days from egg to adult. You can produce large populations, so students in my research laboratory cultivate these flies and keep them in milk bottles with the food at the bottom. Lifespan is about 90 days. So I always tell my students and others that one day in the fruit fly's life is like 1 year in adult life. So to value that fruit fly's life because it's very important. The genetics are very well defined. Anybody who's taken the genetics courses here know that fruit flies were the initial system used to help define a lot of the things we know about genetics today. It's an established model, and we know that the behavior is wired. So we can look at specific neurons and know exactly how they're wired to the behavior. The other thing is that they're very easy to distinguish by eye color, body color, shape of their wings, shape of their abdomens. All these various things are markers that we can use to distinguish the different fruit flies from one another. The other reason why it's so important is because you can model, right, with various different mutations in fruit flies, the various human neurodegenerative diseases. So we can take the individual words, which would be amino acids, peptides, proteins from a human, and actually insert that into a fruit fly. And or there might be an equivalent word in a peptide or protein in Drosophila that translates to the equivalent in a human. So we can model various different diseases with fruit fly strains. And there are some very unique ones. So I'll point out this one, which is really neat. It's called Ether agogo. This is a particular fruit fly that, when exposed to ether, will dance around like a go-go dancer. Okay? And so it develops a particular behavior based on its genetics. Okay, here goes another question for you. I'll give you 30 seconds. Okay, I'll give you less than 30 seconds. This is where when I give a lecture, sometimes I reveal the answer before I ask the question. So I already gave you the answer to this one. I remember this, right? So one day in a fruit fly's life is very important, right? Because it's almost equivalent to the year of a human being, as I mentioned. So we know that the longevity of a human is around 100 years plus or so, depending. So a fruit fly can live, in fact, about 100 days under ideal conditions. But again, you could have answered E, meaning depends on the environment. So if you had an environment that was really rich with food, good lighting, well-kept environment, it could live longer than even 100 days. Okay, so in the final part of my talk, I want to go through the 3 different areas that we study and focus in our lab. And these are all experiments that have been done by the undergraduate and graduate students in my laboratory. The first is developing and optimizing the fruit fly model. So the very first thing we did was to feed the fruit flies this small molecule called BMAA in the fly chow to see if there's any sort of behavioral effects. And I just want to show you a bit of the video that we took. So you can see that this is a female fly that was fed BMAA, and it develops symptoms and signs of what would be resembling of a Parkinsonian patient. One with a lot of tremors, one that cannot right itself. This is also the same fly. And as a comparison, actually when I've sped up the video, we can now look at a control fly. So you can see that this control fly has no problems with keeping itself righted. Okay? And I, in fact, have a very hard time videotaping this fruit fly because it's walking all over. So this is a normal fruit fly. So this is the first indication that there was, in fact, a phenotype from an outward behavior that we could see with the fruit flies fed this BMAA molecule. So we did what we call a simple locomotor disability test, where we tap down the vial of flies that have been fed BMAA, and we count the number that walked to the top of the vial. And so that's one really easy measure for looking at locomotor disability. So that, in a nutshell, is the very preliminary experiments that we did at Fresno State with both undergrads and grad students studying this. Second part was really elucidating this mode of action. So how do you get from feeding the fly this small molecule to it actually developing this odd behavior? And so what I like to often do is to translate how is locomotion controlled, right? So there's some biological action, and there's some chemical reaction, going back to my chemistry, that's related to that particular behavior. And anything that you might define as a chemical reaction might also be defined as an electrical reaction, or transfer electrons, or some sort of difference in the potential that causes a change. And that is a physical output or change in the movement. Here's another question. Okay, so how many neurons does a fruit fly have? Okay, so I'll give you 20 seconds to talk with your neighbor about this one. Okay, let's get a raise of hands. So, how about A, 100 neurons? Okay. B, 10,000 neurons. Okay. C, 100,000. D, 100 million. E, they don't have neurons. All right, there's already one. There's always one in the audience that will say no neurons. So in fact, fruit flies have on the order of 100,000 neurons. Okay, not as many as humans, obviously, but they do have quite a number of neurons in their little fly brain. Okay, so to look at the actual physical output of what we see with the behavior after the fly's been fed, what we can do is what we call electrophysiology on the BMAA-fed fruit fly. So here's your quick fruit fly anatomy, your head, your thorax, and your abdomen back here. We can put electrodes into the fruit fly brain and record the output. And so this is the machine that we use. It's a microscope that has various different micromanipulators, and the actual fly goes right in here at the center. Okay. So believe it or not, we anesthetize temporarily the fly with ether, and we put them in this wax. We record from them, and actually what we can do is save that fly. So we value every single fly that we use as a model system, and we can take that fly back out of that system and put it back into its food source, and it'll live continuously after that. So what have we done? What we can do with these fruit flies is that this is a lot more beyond the chemistry and the biochemistry. This is now looking at fruit fly anatomy. But what we can do is there's a central nervous system or a thoracic ganglion that's connected up to 6 different flight muscles in a fruit fly. And unlike a human, which is multiply innervated, fruit flies are singly innervated. So it's a very nice, simple system that allows us to characterize what's going on with the fruit fly. The other thing is that the major neurotransmitter, or the chemical that a fruit fly uses, is called glutamate. And so when I mentioned the chemistry of BMAA and what it might look like this unusual amino acid, but when it reacts with certain things in the cell, it could also look like glutamate. So the question is, what was going on with what happens when a human, or in this case a fruit fly, ingests BMAA? How does that manifest itself in a behavioral output? So this is neurology or neurochem 101. This shows one neuron here, another neuron here, and we define them as a presynaptic neuron and a postsynaptic neuron. The bits of the chemical packets that are translated and transported from one neuron to another to allow communication are these little red beads here. And these are glutamate. Okay? So they are the neurotransmitter that is communicated from one neuron to another that also results in a muscular output. So what we can do is we can measure what's going on with the electrical output of a neuron. And this is showing a more complicated human neuron, but we can measure the electrical output to know whether or not there's function or dysfunction in that neuron. And so this is showing a recording of what you get when you look at the electrical output from a fruit fly neuron. Okay? So there's this, what we call a fast reaction here. There's a spike up, and there's a graded spike that goes down, and it settles down where there's a slow response. And these are all the things that happen at a receptor protein level. Okay? Again, one of the things that I often talk about when we're looking at biochemical molecules. So what we can do is we've looked at and measured this electrical output in our fruit fly model system, where they've been fed BMAA, this unusual environmental toxin. And what you can see is, in comparison to the initial normal response, these are the responses we're getting from the various different flies fed for multiple days on this environmental neurotoxin. And so you can easily see that there's a difference in electrical output. So what we've translated here is that this electrical output is what manifests in the unusual locomotor behavior and the lack of the ability of the fruit flies to walk. And then the last part is looking at potential therapeutics, or looking at, are there any things that we can help to combat BMAA so it doesn't cause the same deleterious effects as what we see when they are fed or if a person is exposed to it. So these are exact data that have been taken by undergrads in my laboratory. And what we're seeing here is looking at viability on the y-axis and on the x-axis, the various different concentrations of BMAA. And in this case, we're looking at a competing amino acid. So while one word is an unusual word, we can add another word, another amino acid that might compete and cause that unusual BMAA word to be pushed out. Okay? That was our working hypothesis when we started this research. And so what you can see is here, flies can walk normally with normal conditions. With serine, this particular amino acid that we tested, we see that there's no effect as well. When we feed with just the BMAA alone, there is a decrease in the ability of the flies to walk. But if we co-feed BMAA along with serine, we see an abrogation of the effect with just the BMAA alone. And we also see the same thing when we look at serine and locomotor ability. So both in viability, whether or not the fly is alive or dead, as well as locomotor ability, we know that serine is an amino acid that can help abrogate this effect. So to show this a little bit more clearly, the model system we're looking at is if this is a correctly folded protein in this shape, if we had BMAA that might be misincorporated into a sentence, it would form an unusual shape, and that would cause the neurodegenerative disease. And so it's this idea of what happens when a person is exposed to BMAA, when they ingest it, is there some sort of misfolding of a protein that translates into neurodegenerative disease? So one of the things that happened last year after we conducted this fruit fly experiment was our collaborating group also replicated the same experiment in a vertebrate model, in this case, vervets. And this is a study that came out last year where they also modeled the same study in vervets, where they looked at the plaques, they looked at tangles, and they looked at viability. This particular image here shows what happens when you're looking at the various densities of high serine, high BMAA, high serine, plus BMAA, the low control, and then the control here. So basically what we're showing here is that if you have a high amount of the neurotoxin or the environmental neurotoxin, it causes a lot of formation of these dense tangles. But if you co-feed with serine, you see an abrogation of those plaques and tangles. So it's a really interesting and nice, rewarding study to see that our collaborators, using our fruit fly data, were able to progress in their own vertebrate data with the vervets. And at this point, people are now studying what happens with a control group of ALS PDC patients that are being fed low dosages of serine, and perhaps thinking later on down the line what might happen to those patients that have ALS PDC that are fed and exposed to serine over a longer period of time. So that's been one of the most rewarding things about the research that we do in my research lab, is that simple experiment that can be done in the laboratory just next door in Science 1 translates into something that can have an effect and a benefit to others. So it still begs the question of how this is all interrelated between looking at the neuron, these tangles, these plaques, and hopefully we're coming a little bit closer to figuring out what's going on with this particular disease by looking at BMAA, by thinking about protein folding, and by using fruit flies as a way to discover more about what's going on in this particular disease. So I couldn't give a chemistry talk and a description of discovery without doing at least one experiment. So I did want to show you one experiment. And this kind of brings me back full circle to explaining why science and chemistry has been so important to me. It's really this visualization, seeing a chemical reaction and knowing that a chemical reaction occurs and understanding what happens. I did want to bring out a little demonstration of what happens. So a lot of times, when we're studying things in a cellular system or organismal system, we can't necessarily see it, right? But we need to visualize it somehow. And I had initially told you that one of the things that I really loved about the sciences, especially chemistry, was this visual demonstration of what you could see. And so this is one experiment and one demonstration of luminol and luminescence. It's a way in which we can take a chemical reaction and we can visually see it change, okay? And that output, which is shown right here, is that you can have two solutions. One that contains this solution of luminol and copper sulfate and some base, and you can combine it with another solution that contains hydrogen peroxide, which is an oxidant, and we can see an actual chemical reaction take place. So I'm going to actually run this experiment for you to see. But this is part of the reason of why I've loved science so much, is that you can visually see these changes. And this same type of reaction is what you might see on a warm summer day on the East Coast with fireflies. So if you could have the stage lights down. So this is what made me fall in love with science. And I talked about fireworks in the beginning and what you could see. This is what you might see with a firefly. Thank you. And I often like to have demonstrations in the lab. You never know what's going to happen when you're teaching in the classroom, but that's sort of the thrill, the excitement of whether or not it's actually going to work, right? It also means that the students know that you're human, that sometimes it doesn't work, right? In this case, it did work, thankfully. But we can explain the science, right? That this chemical here with peroxide causes some sort of catalytic change with a base to form this excited species that when it goes back down releases light, which is that luminescent color that you saw, which is the same thing that you might see with a firefly, okay? But it's this chemistry that's really been what I've fallen in love with, that you can visually see it, that you know how it reacts, that there are these observations that can be repeated over and over. And so where we're going with our research is to expand looking at fruit flies and looking at this GFP fruit fly. So again, one of the models I always bring in is looking at green fluorescent protein produced by jellyfish. But we can now insert that jellyfish protein into fruit flies, and we can visualize the various different types of neurons. So this is visualizing the cholinergic neurons, and we can make these cultures of muscle cells and neurons that fluoresce green. And so this is where we're going with our research. And I'm hoping that if I ever talk to you, if you ever come up and talk to me on campus, that I can tell you more about these GFP fluorescing fruit flies and how they're going to help us turn out even more research explaining how these various neurodegenerative diseases occur. So I've talked about these three different areas, and I just want to finish by talking about the research students that are in my lab working on these projects. I could not go and give this talk without talking about all the hard work that they do. So this is just a list of all the students that have been in my lab, that are currently in my lab. Some students that have just graduated and been in my lab. And my collaborators at the Institute for Ethnomedicine, Kazuo Ikeda and Jane Koenig, who are my close collaborators at City of Hope down south. Sources of support from the college and the university for providing research opportunities for our students. And then Dr. Krish Krishnan, who's collaborated with me and worked closely on the NMR studies of BMAA. And then lastly, our BMAA Consortium, which is an international group of 50 scientists from all over the world studying this particular disease. Lastly, I just wanted to thank all of you for being here on this evening to hear me talk about my research and the discovery related to BMAA Alzheimer's, Parkinson's, and these various different neurodegenerative diseases. So thank you very much.